All right. So what I'm going to do in this webinar today, I'm going to start by giving you a very brief description of my research focus, the type of research projects I'm involved with, and then I will move into a particular project and take a closer look at genomic analysis I'm currently using in trying to understand the genetic mechanisms controlling palatability or eating satisfaction in beef. And I will focus on 3 different areas. We'll start with the common GWAS study, genome-wide association study. We'll move into genomic prediction and finish up by looking at some different approaches used to move from the GWAS signal and significant SNPs to the underlying biology of the trait. And as we go through these topics, I'm going to try to highlight some of the features available in the SVS software that I use to run some of this analysis. So the main focus of my research program is centered around understanding the genetic control of complex traits. And in my research, I am utilizing the recent advances in the animal genomics field, advances which I think hold great promise on one hand for improving animal production efficiency, but also for enhancing animal products for improved human health. And in terms of specific research projects, over the years, I have been working with a variety of livestock species, with my early focus being on sheep and goats. And these projects included the mapping of QTLs for breeding out of season and milk production in sheep. This was a project I started while at Cornell University. Selection for genetic resistance to internal parasites and genomics of resilience to climatic stressors. These 2 projects started while at Oklahoma State University in collaboration with Langston University. But my main focus over the last several years has been on the healthfulness project in beef, which I'm going to use today for my webinar. And I would also like to mention that this is a project I'm trying to extend here at the University of Florida and combine the meat quality with the nutritional and health value aspect, but in a Brahmin-influenced beef cattle population. I would also like to mention the fact that the ultimate goal in this project is developing genetic tools, which could be implemented in traditional selection programs for ensuring the economic viability and sustainability of the U.S. animal agriculture, whether it is improving nutritional and health value of beef for better human health, or increased milk production, or ability to breed out of season in sheep, improve resilience to climatic stressors. My research is really directed at providing practical industry applications. Now today I'm going to use some data from what became known as the Beef Healthfulness Study. And this study was initiated a number of years ago under Jim Ricci and Dorian Garrick's leadership at Iowa State University to investigate fatty acid and mineral content in beef as possible targets for improving the human healthfulness of beef. The project aimed at assessing the natural variation and understanding the genetic mechanisms underlying healthfulness traits. And the ultimate goal was to develop tools to select for animals that produce more nutritious beef without jeopardizing the eating quality, meaning tenderness and flavor, and without making its production cost prohibitive. And we really started with this idea that genetic evaluation for nutrient composition of beef could result in development of genomic EBVs, which will help selecting for favorable fatty acid profile, lower levels of cholesterol and saturated fat, but also higher concentration of minerals and vitamins. Now, the healthfulness study involves several cohorts representing almost 2,300 predominantly Angus cattle, and these were assessed for a number of traits: growth, carcass and meat quality attributes, including tenderness and sensory information, but also, in addition, extensive phenotyping of traits that might influence the human healthfulness of beef. All the animals were genotyped initially with the Illumina 50K SNP chip, and we also had at our disposal a 5-generation pedigree with almost 6,000 animals. But today I'm going to only focus on the meat quality traits, and this include the Warner-Bratzler shear force and sensory panel attributes like tenderness, juiciness, connective tissue, and flavor. So, first of all, why the special interest in meat quality, or more precisely eating satisfaction? Well, the reason is really this is a very important trait for beef consumers. And if we take a look at the 2011 National Beef Quality Audit, this is a comprehensive survey that evaluates beef industry efforts to improve beef quality. It's conducted every 5 years since 1991, but in 2011, the only quality category that was identified by packers, food service buyers, and retailers for which the sectors are willing to pay a premium was eating satisfaction. And when I say it is eating satisfaction, we really mean tenderness, juiciness, and flavor. So eating satisfaction is a great interest to the beef industry, as improving this trait should lead to increased beef demand. Moreover, if we look at the workshop that followed the audit, 41 industry leaders who represented all sectors of the beef industry participated, and among the top 10 quality challenges that they identified, tenderness and marbling, you can see, were top 3 and 4. And in fact, this is really not a new trend. If we look back since this audit started in 1991, we can see that tenderness, overall palatability, and eating satisfaction always ranked in the top 5 of the quality challenges priorities. Now, at the end of 2011 National Beef Quality Audit, specific recommendations were made to reduce the barriers and improve beef profitability, and this included increased funding to improve eating satisfaction, use of genetics to optimize palatability, but overall the goal was to feed cattle with customer expectations, which will require assisting producers with selection and management techniques to help them produce cattle that feed customer expectations. What are the challenges in delivering the selection and management strategies to producers? Well, they are all related to the fact that we deal with traits that are measured late in life, actually after slaughter, and they are also expensive to measure on a routinely basis. Also, if you take a look at the beef industry today, we are using the USDA grading system, which is essentially based on marbling and maturity, to predict the palatability of meat and communicate it to the consumers. And in this system, a higher quality grade is perceived as an indicator of more tender and palatable meat, which is not always the case. Now, although the USDA grading system has served the industry well, changes in consumers' preferences, limitations in the ability of the system to predict eating quality, but also limited consumer understanding of how the system works are some of the problems associated with the system. So it is also important to point out that, as we all know, beef is an expensive animal protein. What sets it apart are distinct sensory attributes or high palatability. So programs to improve eating experience when consuming beef and the ability to better predict the eating quality level for marketing purposes are critical to increase consumers' confidence that quality expectations are met. So, from that point of view, eating satisfaction is of great interest to the beef industry, as improving these traits should lead to increased beef demand. So how do we define and how do we measure palatability or eating satisfaction? Well, there are a number of measures that can be recorded and used to describe these attributes. These measures include a Warner-Brutzler shear force, tenderness, juiciness, connective tissue, beef flavor, and also a number of off flavors that we can detect. For our study, we collected rib sections from each carcass, and all steaks were aged for 14 days from the harvest date. 2 steaks were actually analyzed, one for Warner-Brutzler shear force. Now, Warner-Brutzler shear force really measures the force required to shear a cooked steak. And for this, we took 6 cores from each steak. They were sheared, and then the average peak load was analyzed. The other steak was used for sensory panel. We had a trained panel of 8 members which evaluated each steak, and then again the average score of all the panelists was analyzed. Juiciness, tenderness, and connective tissue were assessed on a 8-point scale. Flavor and the off flavors were assessed on a 3-point scale. And I just wanted to give you a quick visual of how these traits look in our population. You can see in the table at the bottom right corner the number of animals analyzed for each trait, but also the mean and the standard deviation for each trait. And then I wanted to give you some graphs, the histograms. I think it gives you a good idea of the type of natural variation present in these traits in our Angus population. And remember, that was one of our goals, to see how much variation we have in these traits to start with. But today what I would like to do is take a look at our genomic approaches of addressing this issue of improving the palatability or eating satisfaction, and how do we better predict the eating quality level. And I will touch on 3 different areas: genome-wide association study, genomic prediction, and then at the end I will show you a little bit of how do we move from the GWAS signals closer to the underlying biology of these traits. I will start with the genome-wide association study. And here the goal is really to identify SNPs or chromosomal regions associated with each of the palatability traits: Warner-Brad-Seller shear force, tenderness, juiciness, connective tissue, and flavor. And all these analyses were performed with the SNP and variation suite in Golden Helix, and I'm going to show you how this analysis was done. Now, first of all, SVS has a variety of features to edit, manipulate, and enrich the data. Once the genotype file is imported, a marker map can be applied, and this will allow identification of genomic coordinates of SNPs, but also other genetic features that you have in the spreadsheet. Marker maps are the basis for data visualization in Genome Browse, which I will show you in the next few slides. But what you see in this table, in the top right corner, you can see the number of rows, in our case the number of animals, and the number of columns, or the number of SNPs. In purple is the name of the column chosen as a dependent variable. In this case, I'm showing you here the average Warner-Brad-Seller shear force. And then highlighted in green, you see the marker information, which includes chromosome position, other SNP information. And this is also used later on to add any annotation data, which is a new feature in SVS, but it's a great addition, which makes a lot of the subsequent analysis really a breeze. A number of filtering methods are also available from the Genotype Filtering window, and this allows you to choose thresholds for multiple statistics to filter your SNPs. So you can do it based on the call rate, on the minor allele frequency, but also you have the ability to do Hardy-Weinberg equilibrium filtering if you choose to do so. Now, under the mixed linear model analysis, SVS has a number of options for the association study, and this includes a linear regression, single locus, or multi-locus mixed models. All of them allow correction for relatedness or pedigree structure. There is also an option of pre-computing and saving the genomic relationship matrix, option which actually I found quite helpful because it saves some time if you run many analyses. But you can also use an identity by descent matrix or an identity by state matrix to correct for relatedness. And in addition, all models allow correction for additional fixed effects and covariates. In our case, we use contemporary group as a fixed effect in all the analyses. And then the multi-locus mixed model allows for simple stepwise mixed model regression with forward inclusion, backward elimination of genotypic markers as fixed effect covariates. Now, this is a screenshot of the output generated from the single mixed linear model on the Warner-Brad-Seller shear force. And you can see the type of data that you get out, the p-value, negative log 10 p-value for each marker, a number of other statistics including false discovery rate. At the bottom screen, you see the proportion of variance explained by each marker, call rate, minor and major allele frequency. And then, of course, the first thing that everybody does, right, is plot a negative log 10 p-value to get the Manhattan plot. This in SVS is very easily performed because of the integration with Genome Browse, allows you further visualization and analysis of the genomic data. There are a number of data source library files that can be added to the Manhattan plot, and what I'm showing you here on the slide, I actually have two gene tracks. I have got the gene track from Ensemble and NCBI. And then from this view, you can zoom in into particular regions of interest, start exploring some of the local features, and you really need to zoom in a little bit more to start seeing some of the genes underlying those regions, but you can see now you can read some of the genes. But really, to really be efficient, you need to leave the pretty view of the Manhattan plot and look at the table. So you can get all the feature lists underlying the highlighted or the selected regions that you have, and that's where you find a lot of information. Now, I'm not going to spend a lot of time on these genome-wide association results, but I would still like to show you some of the similarities we get when running Warner-Brotzeler shear force, tenderness, and connective tissue. And these are the three plots, Manhattan plots, that you have in this slide. Now, we were expecting this because these are traits highly correlated. They are measuring similar things, but it is nice to see the similar pattern on the Manhattan plot. It's also nice to see some of the genes that we know are controlling the tenderness popping up as some of our most significant signals in this analysis. And I also need to point out that we've got strong signals also for juiciness and flavor, which is very encouraging, especially for flavor, which I will remind you, this was measured on a 3-point scale, therefore you've got a slightly lower resolution and also a little bit less variation in our population, but a lot of good signals to follow up on. Now I'm going to move now into genomic selection, which provides new opportunities for us as animal breeders. And it is now possible to genotype cattle, right, for at least 50,000 SNPs like we did, estimate the marker effect from a population with SNP genotypes and trait phenotypes, and use them to generate direct genomic values. And with this technology, we now have the ability to select for difficult to improve traits. Those are traits expressed in only one sex, traits expressed late in life, traits require individuals to be sacrificed, or traits which are just too expensive to measure. And palatability traits are a perfect example of this type of traits. They are impractical to improve through traditional selection, but they are ideal candidates for genomic selection if genetic markers that account for a worthwhile proportion of the variation could be identified. Genomic prediction can be performed in SVS either under a Bayesian genomic prediction with the option of the Bayes C or Bayes C pi, or using the GPLOB method, which computes the genomic best linear unbiased predictor of additive genetic merits by sample and of allele substitution effects by marker. The genomic prediction model uses the contribution of each genetic locus to build the model, and it solves for genomic EBV and allele substitution effect. The output generated presents a number of statistics, including a breakdown of the variance components and heritability estimates for the trait. You can get to the allele substitution effects, and you can actually use this similar to the ones that you get in the GWAS. You can plot them in the Genome Browse to generate a very similar plot. And actually what I'm showing you here at the bottom graph is the normalized allele substitution effect value from a Bayes C pi analysis on Warner-Brotzeler shear force, and this actually has a lot less background but shows a similar pattern in terms of the most significant SNPs. And we can also look, so we can run these plots directly in SVS. We can look at the real phenotype. In this case, the real phenotype is the Warner-Brotzeler shear force. and the genomic EBV to get a feel for the correlation between the phenotype and GEBV. Now, the accuracy of the genomic breeding value, of course, is key to the successful application of genomic selection in animal breeding, but cannot really be assessed in the training set. So the goal here was to compare the genomic prediction results and estimate the accuracy of genomic predictions. And I did this in two different ways. One way was to perform a cross-validation in a sample of individuals that are related to those in the training set but are not themselves included in training. And the second approach was to estimate the accuracy, which I'll show you in the next few slides. But for assessing the performance of the genomic prediction using cross-validation, I used a 5-fold cross-validation, and the basic process for running the cross-validation is pretty simple. You randomly divide the data into several equal subsets. In my case, I used 5 sets, and then iteratively create and test predictive models such that each of the subsets is withheld and used for model testing one time, while the remaining subsets are used to train the model. Now, the k-fold cross-validation functionality in SVS is relatively new, but can be used with GBLA, BASE-C, or BASE-Pi. And the function allows you to specify model covariates, prediction method options, the number of prediction iterations or folds, and also stratification variables. So if you want to use a stratification variable, you can ensure that each of the random subsets has a proportional allocation of samples from various subgroups in the data. For example, if you want to stratify by gender in a 5-fold cross-validation, each of the 5 random subsets of samples will have similar numbers of males and females. Now, SVS will give you a summary of the model performance, including measures of correlation between actual and predicted phenotypes for each, for the samples in each fold. It gives you a combined measure of overall performance, and in this screenshot we see the overall results together with individual results from the first 3 folds. We can see the correlation of actual and predicted phenotypes is similar in each instance, with a correlation of about 0.4. The SVS gives prediction output for each iteration of the prediction process, and that includes the allele substitution effect and the predicted phenotypes. And you can see the screenshot. You can get the allele substitution effect calculated in each iteration of the 5-fold cross-validation procedure. You can, of course, visualize the prediction performance. If you want to do an out-of-sample prediction performance, you can plot, for example, the predicted phenotype versus the actual phenotype, which is what I have on this graph on this slide. The predicted phenotype is calculated with that particular animal not included in the model building phase. So that's one way of looking at the prediction, but really a different way of assessing the predictive performance is to estimate the accuracy of the genomic breeding value. And the accuracy is defined as the correlation between the genomic breeding value and the true breeding value. The problem, of course, being we do not know the true breeding values, and the only available information we have to measure the accuracy is the phenotype. So the accuracy of the genomic breeding value was assessed from the genetic correlation between genomic breeding value and the phenotype, and for this I used the bivariate animal model in UMBAN, where the 2 traits that I used were the genomic breeding value and the phenotype. And this resulted in a correlation of 0.59, or approximately 60% accuracy. Now, for the last few minutes of the webinar, I would like to switch from GWAS and genomic prediction into a different area. And the reason for this is that at the end of the day, genomic selection today still treats the genome as a black box. It is really not necessary to understand what is inside the box to make effective selection decisions. But genomic selection could be further improved by integrating pertinent biological information and using efficient methodologies to get from the knowledge of a statistically associated marker to a functional gene variation. And this would move from simply associating sequence variation with distinct phenotypes to a true understanding of the biology of the animal that makes those variations significant. So that will really shed some light on the black box. So I'm going to spend some time talking about how do we move closer to this goal? How do we move from the GWAS signal closer to the underlying biology of the trait? Now, first of all, I need to remind you about the complexity of the system we are working with. We have the outcome we are interested in. We can call it palatability or eating satisfaction, but it can be described by, as I showed you in an opening slide, by different traits or subphenotypes: Warner-Brasseler shear force, tenderness, juiciness, connective tissue, and so forth. For each one of these individual traits or subphenotypes, we have a tremendous amount of data, more than 50,000 SNP effects for each one of the 2,000 plus animals in our population. So this type of setup is perfect for a systems biology approach, where integration of data sets could really allow for a holistic view of the system and discovery of key players. And in this context, we wanted to use SNP association data to guide the inference of gene regulatory networks. This was done using partial correlation and information theory, which is actually an algorithm that was initially developed for gene expression data, but can be adapted to very well to GWAS results. And this multivariate view of the genome-wide association study was termed association weight matrix. The resulting network I'm going to show you in a second is a co-association network comprised of significant pairwise comparisons of gene-centric SNPs. Now, to build the association weight matrix, we start with the genome-wide association on each phenotype in the system. We do need to identify a key trait, and the most significant SNPs for this trait will represent the initial matrix. In my case, I picked the Warner-Brasseler shear force as the key phenotype, and I picked the top, as a starting point, the top 100 SNPs from the Warner-Brasseler shear force. And then we run the PCIT, the partial correlation information theory, which is a Fortran algorithm. It is memory intensive, so I took advantage of our computing power here at the University of Florida using the High Performance Computing Center. But this will identify significant SNPs correlated among multiple subphenotypes and therefore important for regulating the overall phenotype. You get a correlation matrix among all input data points, and then for any correlated pair, you ask the question, does a partial correlation exist to any other data point? And the final output contains direct and partial correlations. So then the rest of the procedure, the matrix of direct and partial correlations, is subjected to some clustering analysis for easy visualization, and this was done in Cytoscape with clustering analysis through the ENCODE plugin. You can annotate the SNPs. That's where annotation is very important. This was done using the bovine UMD 3.1, and then you can enrich the gene ontology terms, identify using David, and then we visualize it using Revigo. Now, the resulting network is a co-association network comprised of significant pairwise comparisons of gene-centric SNPs. And these networks can actually be visualized in a multitude of ways, depending on, of course, how artistically inclined one feels at the moment or how much time you have to kill. But this is an example of one network that was derived based on the top 100 SNPs from the Warner-Brasseler shear force and across all the other phenotypes describing the palatability and eating satisfaction. And just to get you pointed in the right direction here, in the association weight matrix, the nodes are genes represented by associated SNPs included in the matrix, and the edges represent significant co-association between pairs of SNPs. Now, the overall network is really way too big for a comprehensive evaluation. So that's where the ENCODE plugin comes into play, and you can use it to analyze the network, break it into several smaller networks, which then can be evaluated individually to allow for a direct view of the genes involved. And this shows you the top network actually that came from that analysis. It does include a calpain, but it also includes a number of other genes which we can follow up, and actually those are the genes in yellow. The other ones are just SNPs which were not annotated at the time. So to wrap it up, I strongly believe that palatability or eating satisfaction should be very important for the long-term sustainability of the beef industry. But we also need to keep in mind that these are quantitative traits. They are difficult to predict and improve. They are difficult and expensive to measure. They are correlate in life, or they are measured late in life. And collection of palatability phenotypes on a large scale is really not practical. It's really not feasible for the very same reasons, which makes it difficult to consider a genomic prediction or a genomic selection program. However, these and future studies can be successful in identifying QTLs for these traits, and new methods and approaches are already available, as I showed you, to move closer to the functional variants controlling these traits. So with that, I need to thank a lot of people. There were a lot of people that contributed to this work. Special thanks go to Justin Buchanan, my former graduate student at Oklahoma State University. He was instrumental in getting the network analysis up and running. A lot of other faculty and students at OSU were involved with the meat quality measurements. A number of people at Iowa State University, with special thanks going to Jim and Dorian. I also need to mention Dr. John Pollack, who was really the man behind the Healthfulness Project idea, and of course all the financial support that we received over the years. And with that, I think I'm ready for questions. We should have plenty of time.
Thank you so much, Raluca. We are going to answer questions. If you've got some that you haven't entered yet, go ahead and do that in the questions pane. I have a couple quick announcements to allow everyone some time for that. I will have the webcast recording and slides up more than likely today. If not today, tomorrow at the latest. I will go ahead and email everyone the link and slides directly, so look for those in your inbox. As I mentioned at the beginning of the webcast, Raluca was one of our abstract challenge winners, and along with her presentation today, she also did win a year's license to the SVS software. Next December, we will host our 4th annual research competition, so if you're interested in entering, make sure that you sign up to receive updates from us, and you can easily do that on our website. Our next webcast is coming up on August 12th. That will be presented by Vivian Sheehan, who was our other 3rd prize winner in last year's challenge. Dr. Sheehan specializes in sickle cell disease, and she is a professor at Baylor College of Medicine and is part of the Cancer and Hematology Center at Texas Children. Invitations for that will go out in a couple of weeks, so look for those. And with that, we will answer some questions. Let's see, first one here. In GWAS, how do you include relatedness between animals in a situation when there's no pedigree data?
Yes, so actually I think I hope I mentioned this, but I actually did not use the pedigree when I ran the analysis in SVS, but SVS has the ability to calculate the genomic relationship matrix, and this is what I actually did in SVS. So based on the genotypic data that you have on those animals, the genomic relationship matrix is going to be calculated.
Okay, let's see. Did you perform LD pruning?
No, I actually did not. So I just wanted to show you all the different, and I did not show you all, but I just wanted to show you some of the options that you have in SVS. I did not perform LD pruning, and it also, you know, it depends on what your goal is with those analyses. For example, for the network analysis, if you're interested in looking at those associations, you really want all the information that you can get. So I don't want to get rid of SNPs initially. I want to keep them all, and then I'm going to prune them based on those correlations that I find in my system. But if you really want to, so if you're only looking, if you're interested in getting to the very significant SNPs in maybe a GWAS setting, yes, you can perform the LD pruning. I did not, because my goal was not to, you know, it was just to start with all the SNPs I had available and then go through the other analysis.
Okay, how big is the SNP chip as compared to the cattle genome in terms of coverage?
Let's see. Well, it is a 50,000 SNP chip, so that's how big it is. It gives you a pretty good coverage to do all the type of analysis that we've done so far. Of course, nowadays you can go to the 700,000 SNP chip, but in terms of just getting those significant signals that you have, the 50,000 gives you pretty good coverage.
Okay, there are a few more questions on there. I wanted to give you the opportunity to take a look. Or I can just start asking them as well. We've got quite a few moving in.
Yeah.
I'll just start asking. Let's see, here we go. If you're doing Hardy-Weinberg equilibrium, excuse me, I'm tongue-tied, how are you modeling the population structure of your samples? Example, pedigree.
Yeah, so I did show the option of pruning your data based on Hardy-Weinberg, and that's not what I did. So you do have the option in the system, but I did not prune it on the Hardy-Weinberg equilibrium. So
in GWAS, how do you calculate SNPs effects located on sex chromosomes?
There is the option on SVS to do that, and I did not. So for this initial analysis, I did ignore that, but you can. There is an option in SVS where you can specify your sex chromosomes and take that into account.
Okay. If we have 3 to 5 very significant SNPs, is it possible to calculate interaction epistasis between these SNPs?
That's a good question. I do not think at this point in time SVS can look at epistasis. I think, you know, SVS actually is great in taking some of the new approaches and implementing them in SVS as we go. So actually every couple of months there is an update and there's a new analysis that it's possible. But as far as I know, epistasis is not one option that they have right now, but I'm hoping in the future we should be able to do that.
Let's see. You started with 53,000 SNPs and got 51,000 SNPs for final analysis. I wonder, only 2,000 SNPs could not fall in a 0.05 MAF. I'm not sure what that stands
Minor
for.
allele frequency.
Thank you.
Yeah, so the 53,000 SNPs, that's correct, that's what it started with, and then I did some trimming, so I did use the minor allele frequency and I think the call rate as well, but then of course you do lose some SNPs, so when you run the GWAS in SVS, you do get some SNPs trimmed out as well, so that's about right. Depending on your thresholds, of course, you can lose use more. I think I use because as I said, depending on what your goal is for the overall analysis, you can be more stringent. I wasn't very stringent, so I only lost about 2,000 SNPs.
What is the minimum number of animals suitable for a GWAS?
Yes, very good questions. We always ask that question, you know. It's one of those things where the more you have, the better off you are. We had 2,000 animals in this study, actually a little bit more than 2,000, which is great, but it's also very unusual. So very difficult to get to those type of numbers for GWAS. I wouldn't really do it, you know, with less than a couple hundred animals, depending on the trait you're looking at. You're working with a trait with a high heritability, you might be able to get away with fewer animals, but really, you know, you need a good number if you want to get some good results.
Can we pick 100 of the most diverse animals and get the same results?
Well, I guess it depends on what do you mean by 100 most diverse animals. In our population, if you think of maybe picking the extremes, that would be a good question. I did not try that, but I did not try that. I would, you know, if you have a population like ours with, let's say, 2,000 animals, you pick the most diverse, you would still probably have a few hundreds, I assume. Then you might get some good results. But I don't know.
R squared is low in cross-validation phase. Is this a problem?
Well, the R square is low. Yes, it is low, and you know, that's why I said toward the end of my presentation that really you look at those predictions. Yes, they are great, but still your accuracy, it's not exactly where you want it to be, which is why I think that you know, we really need to get to the biology underlying the trait. You really need to have very large populations to get those predictions to be a little bit more accurate. You really need to train them and retrain them, and that's really not feasible, especially for traits, like I said, like palatability or traits which are expensive to measure. They are not measured on a routinely basis. Of course, those are the ones where you want to use the genomic prediction because you want to try to get away from the phenotype, but that accuracy shows you that we are really not in a place where we can do that. That's why it is very important to try to move and get to the genes controlling those traits. If we do have a number of genes controlling the traits, then we can include those, and that would give you some better accuracy.
Let's see. With GWAS, you may get inflated p-values when you have too few cases versus controls. Have you seen that in your analysis? What is an optimal case-to-control ratio?
First of all, so the type of traits that we analyzed so far, we do not have cases versus controls. So we are looking at quantitative traits, even the palatability traits, which are, some of them are categorical. They are still measured on you have more than just a yes or a no. We do not have cases and controls. So so that's really not applicable in this case.
For network analysis, is it best to pick SNPs and exons and perform GWAS?
For network analysis, yeah, so I didn't spend a lot of time on the process of going through those SNPs, but yeah, so what we did, we actually picked SNPs in the gene, first of all, SNPs that fell in a gene, and then depending again on how stringent you want to be, we actually picked even SNPs that were maybe I think we used 2500 kb around the gene, so before or after a gene, Of course, it is best to pick SNPs in exons, but that also brings us back to the coverage of the 50K SNP chip. So I think that if you would actually use the 700K SNP chip to start with, then you can say all I'm going to pick are SNPs which are falling in an exon. But with ours, with the 50K SNP chip, I think you cannot be that stringent and exclude those SNPs. So we actually included first SNPs which were located in genes, but also somewhere around the gene, maybe 1,000 to 2,000 kb from a gene.
Okay, there are lots of kudos and thank yous and not so much questions, but comments letting you know how wonderful your presentation was. I will back them all up on that. Fantastic job. We really appreciate you spending some time with us today and sharing your work. We're going to go ahead and wrap it up. I do see a few questions about the software itself, so we will get in touch with you about those directly. As I mentioned, I'll get the webcast recording and slides up on the website and out to everyone. Thank you so much, Raluca, for sharing your work. We really appreciate it. And I hope everyone out there has a fantastic day.
Great. Thanks, Cheryl, and thanks, everybody.
We'll see you next time.